A very big hello to all my NEET aspirants out there. Welcome you all to day 4 and I hope your preparation game is going on really strong, my dear children. And I hope you're not forgetting to appear for the mock test as well, because it is very important for you to understand with this much time how many questions would you be able to solve and things like that. And if you're searching for a good mock test, not a promotional thing, but yes, telling you we are offering free mock test. If you want, get yourself registered. I think the link might be given in the description box. Okay, so if you go register there, you'll get access to the free mock test. 5 of them are there. Make utilization of that. For 2 of them we have given you video solutions. For the rest of the 3 you would be having solutions in the channel wherever you are going to get the question paper as well. Yeah, and there you have a proper setup in Novo app, my dear children, where timer would be kept and you have to solve questions within that particular time period. It will be really important for you have to have a practice like that. So definitely do not forget to do that. Yeah, and I forgot to tell about this while I was recording for day 3 and day 2. Suddenly I remembered, and I know the days are really less. You should be definitely trying it out. Yeah, and today's session we are going to cover evolution, human reproduction and reproductive health. Most of them the questions are going to come from human reproduction, my dear children. It gives almost 5 questions each year, so we can expect that much coming around from this particular chapter. So if you compare all of them, this is the most important. Then comes evolution, and then towards the end with 2 questions comes reproductive health. Okay, evolution can give you almost 3 questions, 3 to 4 questions. So yes, let's get started with evolution first. So from his experiments, S.L. Miller produced amino acids by mixing the following in a closed flask. What all did he put in the closed flask? So what was he trying to do? He was trying to prove chemical evolution of Earth. What do you mean by that? Ma'am, inorganic things comes together to form organic compounds, and these organic compounds comes in different combination to give rise to a whole living organism, and that is how the world evolved. That is what was said in chemical evolution, and it was put down by Oparin and Haldane. Okay, who proved it? It was S.L. Miller, and S.L. Miller proved this experiment by giving off or by setting up the same situation which was there in the primitive Earth. So he said there was methane, okay, which is represented by CH4. Yeah, he said there is hydrogen gas, which is represented by H2. He said there is ammonia, which is represented by NH3, and he said that there would be reducing atmosphere. That means there is no oxygen, and the temperature was really high. So he kept the temperature around 800 degrees Celsius. Since he was saying there is no oxygen, he set it up in a closed flask. There is no oxygen transport happening, and my dear children, there was water vapor as well. So basically what he did was he put a chamber, he put water for boiling, yeah, and the water boiled, gave down the water vapors in another thing attached to this particular thing. This water vapor went to here, went until this particular position where all the gases were there. These gases along with water vapor in the reducing environment with high amount of temperature, they basically settled down to form amino acids, the organic compounds which is there. Okay, so what all was it? It was methane, hydrogen, ammonia and water vapor. Now those of you who do not know the formula of methane and ammonia get this answer wrong even after knowing if it's methane and ammonia. So CH4 stands for methane, my dear children, and NH3 is for ammonia. So basically your answer would be this, A. Works? Chalo. Next question. Among the following sets of examples for divergent evolution, select the incorrect option. What do you mean by divergent evolution? That means 2 organisms are diverging. They are moving away. But if they are moving away, probably they were at the same point. Yeah. So can I say they are someone who had common ancestors? Yeah. They would be having structural similarity. Yes, my dear children. But they are moving away, and now they would not be having what? Functional similarity. As in structurally they might be same. Structurally within us might be same with them. But the problem is functionally we might be working differently. Okay, example, I'll give you an example. For example, forelimb of man, forelimb of cheetah. We all are mammals, my dear children. Common ancestor, mammal. Yeah, and now we evolved. You have human being and the cheetah. Forelimbs are there, hind limbs are there, same set of bones are there, that is humerus, ulna and radius and all these things is there in us and also in cheetah. But in cheetah they use their forelimbs for running. What do? We dance, we eat, we do whatever we want with our forelimbs. Functions are different? Yes. Structurally is it same? Yes. Do we have common ancestor? Yes. This is divergent evolution. They just diverged, and whatever habitat they landed up in, they evolved, they adapted to that area, which is why functionally we are different. Okay, so here you are asked about incorrect options regarding divergent evolution. So forelimbs of man, bat and cheetah, this is correct. Heart of bat, man and cheetah, this is also correct. We are all mammals, my dear. We have 4-chambered heart, right? Now, brain of bat, man and cheetah, that is also correct. Eye of octopus, bat and man. Octopus, my dear children, is an invertebrate. What is it? It is an invertebrate. Yeah? Agreed? Okay. Now, octopus being an invertebrate and bat and man being mammals, my dear children, and they have eye. In all this organism, eye is for seeing things. Eye is not for anything else. So you see, common function is there. There is what? A common function. And do they share the common ancestry? No, this is mammals, this is invertebrates, this is chordata, this is non-chordata, right? So can I say no common ancestry? No common ancestry. And for divergent elevation, you need to have what? Common ancestry. So this itself is wrong. And common function, that is also wrong. They should not be having common function. You should be having different functions as well. So this is an example of convergent evolution, where something are converging to a point, to a similar point, okay, having similar function. So divergent evolution means your function has to be almost different, at least structurally. A common ancestor should be there, okay. Any one of these or two of these parameters should be matched, okay. Common ancestor should be there, and basically structurally they should be same, functionally obviously they can be different, yeah. But over here it's no common ancestor there and they have common function. This is convergent evolution. This is convergent evolution. And the organs that show divergent evolution, they are known as homologous organs. So HD vision, okay, HD, you can remember like that. And convergent, the organs that show convergent evolution would be what? Analogous. So AC and HD. That's how I remember it. That's how you can remember it as well. Now, which one of the following options gives one correct example each of convergent evolution and divergent evolution? Convergent evolution, eyes of octopus and mammals. Divergent evolution, bones of forelimbs of vertebrates. Convergent evolution, it is eyes of octopus and mammals. Yes, they are converging, they have common function, but they are, let's say, structurally and ancestrally very different. Bones of forelimbs of vertebrates, my dear children. Correct, that is divergent evolution, right? They diverge, which is why all of them are using it for own different purposes. Bat is a mammal, using it the forelimb for flying. Cheetah is an animal, using it for running. We are mammals again, we are using it for having food and dancing and whatever things is there. So basically this is the correct option that matches, right? Now let's see the other ones also. Thorns of bougainvillea and cucurbita. Thorns of bougainvillea and cucurbita, my dear children, they are not showing, it is not thorns of bougainvillea cucurbita. It is thorns of bougainvillea and tendrils of cucurbita. Tendrils of cucurbita. These are two separate things, okay. So thorns of bougainvillea and tendrils of cucurbita, they show divergent evolution. So what I'll tell you how. So basically both these tendrils of this particular thing, that is cucurbita, and the thorn of bougainvillea arose from the same point, that is axillary bud. Axillary bud. So can I say the structural similarity would be same, even though they formed something else later on, but they evolved from the same area, same place, right? So this is an example of divergent. They diverge to different things. One formed tendrils, another formed thorns. So this is divergent evolution, not convergent evolution. Remember that. Now wings of butterflies and birds. Wings, common thing. It is used for flying. Common function. If common function is there, convergent evolution. C for common, C for convergent evolution. Okay, so this is not divergent evolution, wrong. Bones of forelimbs, I told you it will be divergent evolution because structurally we are same, common ancestry is also there. Okay, functionally we are different. Wings of butterfly and birds, I told you again, that is convergent, this should be divergent. Thorns of bougainvillea, divergent. Eyes of octopus and mammals, convergent. So your only answer becomes this one. Cool? Yes. Next, given below 4 statements, each with 1 or 2 blanks. Okay, select the option which currently fills up the blanks in the 2 statements. Wings of butterfly and birds look alike and are the result of what evolution, my dear children? They look alike. Function is same. Function is also same. But structurally, you can break the wings of butterfly like this, but birds, no. Okay, structurally they are not same. So that is convergent evolution. Common function, convergent evolution. So this I should be convergent. I should be convergent. Which all option says I is convergent? Sorry, 1 is convergent. I think, okay, in A 1 is convergent, that is correct. And then again in A 1 is convergent, this is also correct. Any another thing? No. Okay, fine. So we have these 2 options. Now we can look around it, okay. Now it's talking about D over here. Let's see D. Directly go to D. According to Darwin, evolution took place due to 1 and 2 of the fittest. Due to what? Small variation, my dear children, and what? Survival of the fittest. So 1 will be small variation and 2 will be survival. Is that correct? Yes, I already got my answer without going ahead with any of the things. So you have to spot the correct answer from here, then go here, then see the other options. If it is matching, then your correct answer is here. You do not have to linger around anywhere. Yeah. Now what is the problem with other option? Let's see that now. Okay. So suppose Miller showed HCH4, H2 and NH3 and dash when exposed to electric discharge in a flask resulted in the formation of dash. Resulted in the formation of amino acids, my dear children. And what is this dash? Water vapor. Okay. These were the 4 things that were used. It was water vapor. So basically 1 should be water vapor. B1 should be water vapor, 2 should be amino acids. Okay, what about here? C. What about C? Vermiform appendix is dash organ and dash evidence of evolution. Is dash organ what organ, my dear children? Vestigial organ. But here it is written rudimentary organ. That is wrong. Again, you get it? That is why all the statements are being wrong apart from this. Only this one is correct. Over here, oxygen is written for this. Oxygen was not being used. It was an aerobic situation that was being created, right? So your only answer is A. A for apple. Next, variations caused by mutations as proposed by Hugo de Vries are. So, Hugo de Vries was working on primrose and he was the person who said that the variations that you see over here, it's not just random, it's basically, it's a random process, it can happen randomly and this is known as mutation. This is known as what? Mutation. And mutations are random and they are directionless. That means anything can happen. Good mutation can happen, bad mutation can happen. It can favor evolution, it cannot favor evolution. Anything can happen. That's what Hugo de Vries said. Contradictory to it, initially who said something? Darwin was saying that variation is occurring, I don't know how and all, but these variations which are there, they are small variations and basically they are directional, they generally favor evolution. But it's not like that, it is random, it is the variations that you see, it is a result of mutation and mutations are random that occurs and they are directionless, and that was said by Hugo de Vries. So your answer would become random and directionless. Cool? Yes. Next, a gene locus has 2 alleles, capital A and small a. Fine. If the frequency of dominant allele is 0.4, okay, then what will be the frequency of homozygous dominant, heterozygous and homozygous recessive individuals in the population? So A is 0.4. That means you're talking about, you might have studied the Hardy-Weinberg principle, right? So in that, P square plus 2PQ plus Q square is equals to 1. Where? If you're talking about P square, you're talking about capital A, capital A. If you're talking about Q square, you're talking about small a, small a. If you're talking about 2PQ, basically you're talking about capital A, small a, that is heterozygous condition, right? So here P's value is, P's value is given, okay, not P square's value, P's value is given and that is 0.4. Right. Now what we have to find out? Frequency of homozygous dominant. That means we have to find AA. So what do? We do P square. We do P square. So 0.4 whole square is what? 0.16. Okay. Now what? You want heterozygous. Okay. The frequency of heterozygous. For that you have to find A also, my dear, small a also. How do you find that? If you know P, you can find it like this. P plus Q is equals to 1. P is 0.4. Then P plus Q is equals to 1. Q will be what? 1 minus 0.4. Q will be 0.6. Q will be 0.6. So over here plus 2, capital A is what? 0.4. Right, and small a is what? 0.6, right? The whole thing, when you get 0.24 into 2 will be 0.48. This is heterozygous frequency. Yeah, now talking about homozygous recessive. Homozygous recessive is this, yes, this is the recessive allele. How do you find out? Q square you do, that is 0.36. So your answer should be 0.16, 0.48 and 0.36. 0.16 that is AA, 0.48 that is capital A small a and 0.36 that is small a small a. Okay, always remember the frequency can be different. Okay, do not think that it is a recessive allele, how can the frequency be more? No, that means recessive allele is more, short people are more or whatever is there. Recessive things are more there. That is why the frequency is more. You don't have to be concerned that this is recessive, this is dominant, how can the number be like this? No, do not have that concern. Okay, works. And always you have to multiply it with 2. Heterozygous, for heterozygous you have to multiply it with 2 according to the equation which is given out here. Works? Works. Chalo. Next question. In a species, the weight of a newborn ranges from, weight of newborn ranges from 2 to 5 kgs. Okay. Now, 97% of the newborn with an average weight 3 to 3.3 kg, they survived. Okay, what about the rest of the organisms? Basically the one that had 2 to 2.5, they were not able to survive, and also the one that had 4.5 to 5 they were also not able to survive. That means what type of natural selection are they showing? They are showing stabilizing selection, where the graph would look something like this. This is the graph. When you, if at all the question was that this particular organism, say babies with this much kg and this much kg were chosen, but no average were found, then it will be like this. This is known as disruptive. Two extremes are chosen. Okay, now if only one type of babies were there, okay, that is 4.5 to 5 kg children were surviving. So in that case, it will be one extreme that will be chosen. That is known as directional. So you have in between, then it is stabilizing. If it is cut into two extremes are chosen, then it is disruptive. And if it is moving towards one side, only one extreme character is being chosen, it is known as directional. So over here, it is stabilizing because the median range is being chosen. The average one is being chosen. Works? Okay. Next, genetic drift operates in. What do you mean by genetic drift? Genetic means sudden change in the population frequency, in the genetic frequency. For example, my dear children, I had green beetle over here. And I also had white beetle. So frequency of green beetle is more than white. Agreed? Agreed? Now what happened? All of a sudden, an elephant came. All of a sudden, an elephant came and stamped on this. Looks like a frog's leg, but still, yeah. So elephant came and stamped on it. All the green ones went off. Again, the frequency of the green ones gone. It's changed completely. This is occurring by chance, right? This is known as genetic drift. Genetic drift is when the population frequency or genetic frequency of some alleles is changing by chance in a population, and it will be operating only in a small isolated population, not in a bigger population. In bigger population, you'll never mind, there might be more other green beetles out there. You get it? In a small isolated population, this works. So please remember, genetic drift is for small isolation population and it is by chance. And this is, genetic drift is something that breaks the Hardy-Weinberg law. Hardy-Weinberg law says that the allelic frequency will be same after generation after generation. So there are 5 factors which say that it cannot be possible like this. Allelic frequency always changes. One is gene flow. When some organism come over here, some organism go over there, frequency is changing. Yeah. Second thing is genetic drift. Third thing is mutation. Mutation can happen anytime. The fourth thing is genetic recombination, my dear children. While sexual reproduction is happening, new combination can be formed and new frequencies, new alleles can be formed. The frequency of the older ones can be changed, obviously. And the last one was natural selection. Nature might be selecting some organism over the other ones. So anyway, the frequency could be changed. It cannot be same generation after generation. So your answer for this question would be A. Now, dinosaurs dominated the world in which of the following geological eras? Cenozoic, Jurassic, Mesozoic, Devonian. What is your answer? Your answer should be Jurassic. You know Jurassic Park? Okay, you know Jurassic Park. If you're talking, look, listen. In the Mesozoic era comes Jurassic, Triassic and all these things, okay? So please understand that, but if you are very specific, you're very specifically asking, you can go for Jurassic. Dinosaurs dominated the world in which of the following geological eras? Jurassic. Works? Now, match the hominids with their correct brain size. So first of all, you should be understanding this one very important thing in evolution, and that is, what is the pattern of evolution? First thing, first men who came, okay, or the first origin of man, if you see, there is Dryopithecus and Ramapithecus. Dryopithecus were more ape-like. Ramapithecus, Ram, they were more man-like, okay. Now from Ramapithecus came Australopithecines. From Australopithecines came the first human-like hominid, that is Homo habilis, Habibi, okay. Homo habilis was the first human-like hominid, my dear children, that we had, and they generally ate fruits. They never touched meat and things like that. They only ate, they were pure vegetarians, okay. Now their brain capacity was around 650 to 800 cranial capacity, that is CC, okay, 650 to 800. Now from Homo habilis came Homo erectus. Homo erectus started eating meat and all, okay, and their brain capacity increased also. So it was 650 to 800 cc first. Now in Homo erectus it became 900 cc. Cool. Now from Homo erectus you got two things, okay. One was Homo neanderthalensis, which came before, okay, and they have the biggest amount of brain capacity, and that is 1400 cc. You know what, they were kind of emotional, they buried their dead, they covered their body and things like that. Neanderthal man with 1400 cc cranial capacity, okay. I told you two of the organisms came, one is Neanderthal man and then came Homo sapiens, which is a recent one, that is us. Our brain capacity is comparatively lower than the Neanderthal man, so it was 1350 cc. So D goes with 2. D goes with 2. So let's see. Homo habilis. A goes with 3. A goes with 3. I have so many options like that. So probably I should be looking for D. Homo sapiens goes with 2. Homo sapiens goes with 2. Again, I have so many options like that. Oh my god. Okay. Let's talk about C. Homo erectus, it goes with 900 cc, that is 1. C goes with 1. I have, oh my god. Okay. And then in that case probably, okay, chalo. Homo habilis, let's start from the beginning. So A goes with 3, agreed? A goes with 3, I have 3 options. Let's cut this option. Okay. Now D goes with 2. D goes with 2, I have 2 options. Let's cut this. Okay. Now Homo neanderthalensis, it goes with 4. That is B goes with 4. B goes with 4. I just have 1 option and this becomes my answer. Yes. Okay. And with that, we complete evolution as well. Few more important things. What was the connecting link between fishes and amphibians? It was, tell me, fast. It was lobe fins or coelacanth. Okay. Now, what was the connecting link between, the reptiles and the mammals? They were shrews. It was shrews, which was a connecting link. There are so many other things that is there in evolution, my dear children. If you're facing difficulty in understanding certain concepts in evolution, you can put it down in the comment section. I always tell that I become really happy whenever you put some doubt or whenever you say something good. Okay. I really appreciate your efforts of participating in this class properly. So please do that. I'll be really happy, and I'll do my own research, try to find the answers as soon as possible and give it to you. Fine. So please do that, and please refer to your NCERT. Very important. Solve as many questions as you can. I'm just here to at least give you a little amount of help in ways possible, right? So yes. Now going ahead with the next chapter, which is a very important chapter, that is human reproduction, my dear children. Let's get into it. So, first question, select the correct sequence for transport of sperm cells in male reproductive system. In male reproductive system, my dear children, what do you have? You have testes. In the testes, you have what? Testicular lobules. Agreed? This is the testicular lobules. Inside that, you have seminiferous tubules. Agreed? These seminiferous tubules are they're gonna come out with the help of tubuli recti, which is not there in your NCERT. Let's go ahead. You have rete testis. You have what? Rete testis. This is the first thing that you have. After seminiferous tubule, you have tubuli recti, seminiferous tubule, and then tubuli recti, and then rete testis. Rete testis, from here they have to exit. So vasa efferentia, E for exit, E for efferentia. Okay, they're exiting, but they have to make a stop over here, and they make coiled structure over here, which is known as, first of all, let me write vasa efferentia, and then you have epididymis, which is actually 6 meters long if you keep it open like that. Yeah, and then from epididymis you have a connecting link that is known as vas deferens. Okay, so what is your answer? This one. And tubuli recti will not be there, so I'll say this is second one, this is third one, this is fourth one, and this should be the fifth one. Okay, so seminiferous tubules, rete testis, vasa efferentia, epididymis, vas deferens. Okay, now after vas deferens, what do you have, my dear children? Vas deferens basically opens up into the urethra, but before opening up, it joins up with something. It joins up with something else. Listen. So what happens is that when we reach the uterus, right? Sorry, not the uterus. Oh my god, I'm so sorry. What do you call it? Urinary bladder. Yes. I brought uterus in the case of males. I'm so sorry. So urinary bladder, when you reach the urinary bladder, you receive a secretion from seminal vesicle, which is one of the glands. Okay. You receive the secretion. Together, both of them are going to open into ejaculatory duct. This ejaculatory duct opens into what? Urethra. Okay. Urethra. So it looks something like this. This is the urinary bladder with the urethra, right? Now here comes your vas deferens, and here comes the secretion from the seminal vesicle, and together they form the ejaculatory duct, which will be opening up over here into the urethra. The urethra, if you see the extension of it goes, the opening of the urethra is known as urethral meatus. So your answer is C. Okay. Next, Sertoli cells are found in, where do you find Sertoli cells? I told you there are testicular lobules over here, my dear children. Okay, and inside the testicular lobules, what do you have? You have seminiferous tubules. If you just zoom this picture in, it will look like something like this. So this is one seminiferous tubule. This is another seminiferous tubule. Inside the seminiferous tubules, you are making the sperms, and for sperms you need two things. One is the spermatogonia, which is going to later divide and form the sperms, and to nourish this spermatogonia you have Sertoli cells, or you also call them nurse cells. They are going to provide nourishment. Where is it, children? Inside the seminiferous tubules. Okay, now I'll tell you one more information. Outside the seminiferous tubule, the space that you see, it is known as interstitial space, and over here you find something that is known as Leydig cells. It is known as Leydig cells. Fine. Now, over here, Sertoli cells are found in ovaries and secretes progesterone, adrenal cortex and secretes adrenaline, seminiferous tubules and provide nutrition to germ cells, pancreas and secrete cholecystokinin. No, my dear children, your answer is this. And I was talking about Leydig cells, which is there in between the seminiferous tubules, right? What do they do? What does the Leydig cell do? Leydig cells releases the hormone, the male sex hormone, androgens. Famous one, testosterone. Okay, please remember that. And this testosterone is very important for the formation of sperm. So basically, the testosterone gets entry into the seminiferous tubules and then do the action over there. Next, seminal plasma is rich in humans is rich in. What is seminal plasma? Ma'am, there are three secretions. Okay, there are three secretions. One, as I told you, suppose this is the urinary bladder. You got the vas deferens from here, both the side, right? And then there is seminal vesicle. This is what? Seminal vesicle. Seminal vesicle gives fructose. What? Fructose is sugar. It provides fructose to the sperm which are coming because they need energy. Now going ahead, both of them are coming, okay? Now they are surrounded by another gland. This is known as prostate gland. Very important. It provides calcium. Calcium is very important for fertilization, okay, fertilization to happen. And apart from that, they also give certain enzymes. These enzymes are also very much important, okay. These two things by prostate glands. Now again it goes ahead, okay. So over here, my dear children, before it enters into the penis area, at this particular position there are small bulb-like glands which are known as bulbourethral gland, or you also call it as Cowper's gland. And what exactly happens over here? They help in lubrication. They help in lubrication. Now all these secretions, my dear children, together it will be known as seminal plasma. Fine. Now, seminal plasma in humans is rich in what? Fructose and calcium, but has no enzymes. It is a wrong statement because it has enzymes. Glucose and certain enzymes but has no calcium. Wrong. Fructose and certain enzymes but poor in calcium. Wrong. Fructose, calcium and certain enzymes. Correct. So hence D becomes your answer. Next, given below is a diagrammatic sketch of a portion of human male reproductive system. Select the correct set of names of the parts labeled as A, B, C and D. So I just told you this is vas deferens. A would be what? Vas deferens. Over here you have seminal vesicle, and then you have prostate gland, and then the small choo-choo thing that you see is bulbourethral gland. So A is vas deferens, B is seminal vesicle, C is prostate, and D is bulbourethral gland. Cool? Next, meiotic division of the secondary oocyte is completed when? When is meiotic division of secondary oocyte completed? You know, secondary oocyte is stuck, okay, in some stage, okay, metaphase stage, okay, and it is just there, metaphase stage of metaphase 2 of meiosis 2, right? So what happens over here in this particular thing is that it is not yet completed the division. It is just in metaphase, right? It has to go ahead with anaphase, telophase and all of this thing. When does it finally complete its division and give rise to the ovum and also the second polar body? Prior to ovulation? While ovulation is happening, while the graphene, inside the graphene follicle you have the secondary oocyte being safe, okay? So during ovulation the graphene follicle will be bursting and the secondary oocyte is the one that is being released. And this secondary oocyte which is there, the sperm will be coming and trying to fertilize it. When at the time of fusion of sperm with an ovum, that is the point where actually it will complete its division and give rise to one ovum and the second polar body. Always remember that when it actually comes and fuses, not with the zona pellucida, but get inside and fuses with the plasma membrane of the secondary oocyte, my dear children. So prior to ovulation, why do you think the secondary oocyte will be dividing completely? At the time of copulation, no. After zygote formation, or for zygote formation, you need ovum, no. So before that, it has to happen. So at the time of fusion of sperm with an ovum, correct. D becomes your answer. Now, the difference between spermiogenesis and spermiation is what? What do you mean by spermiogenesis? Spermiogenesis means, I'll tell you. So, for example, my dear children, just give me a quick still second. Okay, so the story goes like this. You have spermatogonia, okay? Spermatogonia are the cells which will be giving rise to sperms later on. So some of them get differentiated and they form primary spermatocyte. What do they form? Primary spermatocyte. Okay, and what exactly happens to the primary spermatocyte, my dear children? This primary spermatocyte, which is in 2N stage, it will undergo meiosis, divide and form 2 secondary spermatocytes. Agreed? Again it will divide because meiosis happens in 2 stages. Again it will divide. You get 1, 2, 3, 4 cells. Each haploid, 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 haploid. Okay? These are known as spermatids. These are known as spermatids. Now what will happen is that they are immature, they are round, they are globular. You want to elongated cells, right? So you have to make certain structural changes and things like that. This process of conversion of spermatids to the mature form, that is spermatozoa, is known as spermiogenesis. Okay. Now this spermatozoa has to leave the seminiferous tubule and go. This process is known as spermiation, aviation, you can remember like that. They leave the seminiferous tubule and try to go. That is known as spermiation. So spermiogenesis is this, spermiation is going, yeah, and also this whole process is known as spermatogenesis, formation of sperms. Now the difference between spermiogenesis and spermiation is what? In spermiogenesis, spermatids are formed, spermatozoa is formed. Wrong. In spermiogenesis, spermatozoa are formed, while in spermiation, spermatids are formed. Spermiation means going, okay. In spermiogenesis, spermatozoa from Sertoli cells are released into the cavity of the seminiferous tubules. No. In spermiogenesis, spermatozoa are formed, while in spermiation, spermatozoa are released from the Sertoli cells into the cavity of what? The seminiferous tubules. So this becomes your answer, okay. Yes. Next. Which of the following layers in an antral follicle is acellular? That means it is not made up of cells. Which of the following layers in an antral follicle is acellular, my dear children? It would be what? So basically, you know there is a Graafian follicle. And in Graafian follicle, one big thing happens is that inside that you have the secondary oocyte, right? The secondary oocyte is going to form a layer around it, and this layer that you see is known as zona pellucida. This is known as zona pellucida. Outside the zona pellucida, my dear children, you will be having certain cellular layers which is known as corona radiata. That is different, that is cellular. But this is something that is made by the secondary oocyte itself, and that is known as zona pellucida, which is absolutely acellular. So which of these following layers in an antral follicle, and outside that you will be having so many granulosa cells and things like that, okay, and the interna, externa, all these things. So thecas. So stroma, zona pellucida, granulosa, theca, interna. What is your answer for this question? Your answer is zona pellucida. Okay, next question. The figure shows a section of human ovary. Select the option which gives the correct identification of either A or B with function or characteristics. So basically A is this and over here B is there. So my dear children, you have primary follicles containing the primary oocyte over here. They form the secondary follicles and then the tertiary follicles. It forms Graafian follicle up over here. The Graafian follicle will be rupturing, giving the secondary oocyte. Whatever is left behind is known as corpus luteum. So primary follicle, secondary follicle, tertiary follicle, Graafian follicle, it ruptures, gives out secondary oocyte. Whatever is left behind is corpus luteum. And what does corpus luteum do? Corpus luteum is responsible for the release of 2 hormones, but one majorly. 2 hormones, that is progesterone and estrogen, yeah. The major hormone is progesterone, and progesterone is something that maintains the endometrium lining, my dear. Together they perform, but major role is given to progesterone. So always remember it. So corpus luteum does that thing. So over here B is given as corpus luteum and it secretes progesterone. First option itself seems to be correct. Now A is tertiary follicle. A cannot be tertiary follicle, wrong. B is corpus luteum and secrete estrogen, correct. But what is more correct? This one, I told you, primary function is this, yeah. And A is primary oocyte. It is in prophase 1 of meiotic division, my dear children. So apparently it would have been, if it was said as primary follicle, would have made more sense. And politically, if you see, this is more correct, so I'll go with this. Okay, your answer will be A. Next, which one of the following statement is false in respect to viability of mammalian sperms? So, sperm is viable for only up to 24 hours. You have to find the false one, that is the incorrect one. Do you think sperm is viable only up to 24 hours? Wrong. Wrong. It can survive in the body for almost 5 days. So, this is a very wrong statement. Okay, so survival of sperm depends on the pH. 24 hours means 1 day, right? Survival of this sperm depends on the pH of the medium and is more active in alkaline medium. Correct, that is true. Viability of sperm is determined by its motility. Yes, obviously it has to move and then only it will be viable. We can understand the fertilizing capacity as well, yeah. The sperm must be concentrated in a thick suspension, yes. That is seminal plasma plus sperm becomes the semen. So that is also correct because that has all the essential things needed for it, yeah. So this is the statement which is totally incorrect. Works? Yes. Next, menstrual flow occurs due to lack of. Why do you think menstruation occurs? Why do you think endometrial lining sheds down? I told you there is one hormone that is very, very important in the maintenance of endometrium lining, and that is what? Progesterone, which is made by the corpus luteum. Now, suppose that hormone is not there. What will happen? Basically, there will be menstrual flow that will be happening. So, can I say C is the answer for this question? I can say that. Guys, just give me a second. I'll be back. Yes, going ahead. So question number 12. The secretory phase in the human menstrual cycle is also called. What do you call the secretory phase? So my dear children, understand you have certain phases. First one is the menstrual phase where the menstrual flow happens. That is the endometrium will be shutting down. It happens for almost 1 to 5 days. Yeah. Then going ahead, what do you have? You have, this is menstrual phase. Okay, then what happens? The body is given one more chance. Yeah, follicles regenerate. So I'll call this follicular phase, and I also call it proliferative phase. Why, ma'am? Because over here the endometrium is trying to reform with the help of estrogen. Estrogen is always there. Yeah, so that happens in follicular phase. Then here, almost on the 14th day of the cycle. On the 14th day of the cycle, what will happen? There will be a spike and there will be ovulation happen. So I will call this as ovulatory phase. This is ovulatory phase. Ovulation happened, graphene follicle ruptured, you gave the secondary oocyte. What is left behind? You have the corpus luteum, and this corpus luteum basically is going to secrete progesterone, which is why it is going to, thicken more. Now it is progesterone era. It's not just estrogen, it is also progesterone over here. Both of them combinedly thickens, but major role is done by this guy. So this is known as luteal phase. Why luteal phase? Because corpus luteum is being formed and there is a hormone LH, like luteinizing hormone, which maintains the corpus luteum, right? And also it is known as secretory phase. Why, ma'am? Because over here this progesterone, when it thickens the endometrium, it also makes sure that the endometrium is secreting certain very important things. It is what? Secreting certain very important things like glycogen. Suppose fertilization happened, fertilization happened and the implantation occurs, my dear children, it should be able to provide nutrition, right? And that nutrition is the form of glycogen. So which is why it is also known as secretory phase. So this is 14th day where ovulation happened. Now more 13 to 14 days would be your luteal phase, and then again, so total 14 plus 14 almost 28 days that is there. For people the thing differs, it changes, for some cycle is 31, 30 days and things like that, but this is the general thing that you need to know. So the secretory phase in the human menstrual cycle is also called as luteal phase and lasts for about 6 days. No, Follicular phase and lasts for about 6 days? No. Luteal phase and lasts for about 13 days. Yeah, almost. I told you, it's not exact number that we have over here. So, almost lasts for about 13 days. Follicular phase and lasts for about 13 days? No. Follicular phase is proliferative phase. Always remember, when follicles are generating, no. When you are making the primary follicle turn into secondary and tertiary, the follicles, the granular cells around here, they are releasing some hormone, that is estrogen, which will be making things up over here. Okay, so your answer should be C. Now, going ahead, which one of the following is the correct matching of the events occurring during menstrual cycle? Proliferative phase, what happens during this particular phase? I just told you right now, but let's see. So, rapid regeneration of myometrium and maturation of Graafian follicle, which one is correct matching? Wrong, because in proliferative phase, what is happening, my dear children? The endometrium is reforming again, or basically one more thing happens, that is rapid regeneration of not myometrium, it should be endometrium. Myometrium is the muscular layer. Okay, but maturation of Graafian follicle happens over here because it is also known as follicular phase. So, this part of the thing is wrong. So, this is wrong. Secretory phase, also known as luteal phase, development of corpus luteum and increased secretion of progesterone, correct. Menstruation is breakdown of myometrium and ovum not fertilized. Arey baba, yeah, ovum is not fertilized, but breakdown of myometrium, it is a muscular layer. Why are you taking myometrium over here? It should be endometrium. Again, wrong statement. Because some children do not see, they just go and attempt metrium. They see metrium and they go and attempt. Please don't do that mistake. Now, ovulation. LH and FSH attain peak level and sharp fall in the secretion of progesterone. Ovulation, yes, there is LH and FSH that attains peak level, that is for sure. It is specifically LH that attains, yeah. And what happens at the same time when LH is attaining peak level, there is ovulation happening, and because of this, there should be increase in progesterone. Why? Because LH increased, Graafian follicle ruptured, secondary oocyte released, Graafian follicle transformed into corpus luteum releases progesterone. Progesterone basically increases, not decreases. So, this is wrong. Okay, so what is correct? Only B option would be the correct option. Going ahead, which part of the ovary in mammals acts as an endocrine gland after ovulation? Which part of the ovary in mammals acts as an endocrine gland, my dear children, after ovulation, after ovum is being released, or basically secondary oocyte? Just give me a second. Oh my god, I got scared. Secondary oocyte is being released. Is it stroma? Is it germinal epithelium? Is it vitelline membrane? Is it Graafian follicle? Is the Graafian follicle the mature follicle? When it ruptures, secondary oocyte, what is left behind? I told you several times. Corpus luteum, what does it release? Progesterone. Isn't it acting like an endocrine gland? Yeah, because it is releasing a hormone right there. So, can I say the answer for this question is Graafian follicle? Yes, ma'am, you can say that. Yes, I will say that. This is the answer. Okay, going ahead. Identify the human developmental stage shown as well as the related right place of its occurrence in the normal pregnant woman, and select the right option for the two together. Developmental site of occurrence. Okay, fine. My dear children, what is this thing that you see? What is this stage? Wherever you see a big cavity up over here, just understand this is a blastocyst stage. Blastocyst stage. With this thing that you see, the cavity that you see is known as blastocele. This is known as blastocele. Now, the outer layer of cells that you see is known as trophoblast. The inner layer of cells that you see, it is known as inner cell mass. So, the trophoblast layer that you see will be slowly going and implanting into the endometrium. Yeah, suppose it is implanting. It will help in the formation of placenta later on. What happens in the inner cell mass, it is the one that will actually form the embryo later on. Okay, this forms the embryo later on, this goes and attaches over there. Always remember. So, it is not late morula, it is not blastula, it is blastocyst stage. And where is it going and touching? On the uterine wall, that is the endometrium. So, this is the answer. Cool. Now, what is true for cleavage? What do you mean by cleavage? As in, you have the zygote, yeah, sperm and egg fuses, you get the zygote. The zygote is single celled, it has to divide into 2, then into 4, then into 8, then into 16 and things like that. What exactly happens over here? Whenever this big cell divides, don't you think the cytoplasm over here is going to decrease? The size of the cell. The size of the cell, my dear children, it is going to decrease, right? So, what is true for cleavage? Size of embryo increases. Okay, size of cells decreases. Yes, for sure. Okay, size of cells increases, Size of embryo decreases? No. It is size of cells that decreases, my dear children. The cell's size would be decreasing. This is apt. Okay, the size of embryo increases, you might think that is also correct, ma'am. But if we talk about cleavage in general, my dear children, isn't the cell size decreasing? So there is something that is more apt over here. You should be going ahead with that, right? So size of the cells would be decreasing because there is cleavage happening up over here. What about the DNA? DNA would be more. So initially you just have one cell, one DNA, one copy of DNA. Now you have 2, then you have 4. So DNA amount would be increasing, but the size of the cell is going to decrease. Okay. Now hormones secreted by the placenta to maintain pregnancy are placenta and the hormones. There are 4 hormones released by placenta. Human chorionic gonadotropin, human placental lactogen, progesterone and estrogen. You might think, ma'am, progesterone and estrogen released by ovary also, right? Yes, but during pregnancy, placenta takes up this role and releases it. Okay, please remember that. Please remember that. Yeah. And apart from that, in your book there are certain other hormones being mentioned released during pregnancy. What are those? Relaxin by ovary, but the later phases of pregnancy. Here the question is, it is released by placenta, and not only by placenta, it maintains pregnancy, my dear children. So what are those things? HCG, HPL, progesterone and estrogen. This is very much important for the maintenance of pregnancy. Relaxin, which is released by the ovary, it is there for parturition to happen properly, for making relaxation of the muscles around there, for parturition, for baby to move out properly. So it is released during later phase of pregnancy. Now one more question comes, what are the hormones which increases in many folds during pregnancy? That means they are there in the body, but during pregnancy they increase in many folds. Thyroxine, cortisol, all those things. Okay, please remember those as well. Yeah. So this becomes your answer. Now 18th, ectopic pregnancies are referred to as. What do you mean by ectopic pregnancies? Ectopic pregnancy means what? So whenever pregnancy is happening at elsewhere place apart from the uterus, we call this as ectopic pregnancy. For example, this fallopian tube, yeah, there if the baby is developing, it is example of what? Ectopic pregnancy. It is not safe for both the mother and the child. So implantation of defective embryo in the uterus, no. Pregnancies terminated due to hormonal imbalance, no. Pregnancies with genetic abnormalities, no. Implantation of embryo at the site other than the uterus, yes. And that is really, really risky, not risky. Next, the first movements of the fetus and the appearance of hair on its head are usually observed during which month of pregnancy? I love explaining this thing. Okay, so there are few months put in your NCERT, you have to remember them. First month, what happens after first month? Heartbeat, tuck, 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 tuck. When someone dies, you check the heartbeat only, pulse rate and things like that. Similarly, the life also, you get to know that there is life by checking the heartbeat after first month. Okay, second month, you have got limbs, you have got digits. Okay, second month, you have got limbs and you have got digits. Okay, so you can remember 2 limbs we have, so that's why it is second month. After second month you have that, and also after the second month you will be having certain organ system which is formed. External genital organs also will be formed during that particular time. Okay, now third month, what happens during the third month, my dear children? It's not given in your NCERT. Let's drop it. Directly it goes to 5th month. What happens in 5th month? Baby says that, God, you did whatever you want. Now please do something that I want. I want hair on my head and I also want to move around. So that happens in the 5th month, first movement of the baby, and also hair on the head, 5th month. Okay, now 6th month. By the end of the 6th month, what will happen? Basically, eyelids will be formed, eyelashes will be there. Eyelids are separated, eyelids will be separated, eyelashes will be formed, and also there will be hair on the body. There will be hair on the body after the 6th month. So hair on the body, hair on the head is different thing. Okay, please remember that. Hair on the head after 5th month. Hair on the body after 6th month, along with eyelashes and also eyelid separation. So over here, the first movement of the fetus and appearance of hair on its head is usually observed during which month? It will be 5th month. B becomes your answer for this question. Okay. Next, the fetal ejection reflex in humans triggers the release of. The fetal ejection reflex in humans triggers the release of what, my dear children? What do you mean by fetal ejection reflex? The baby is trying to move out. Okay, what is happening, my dear children? The baby is trying to move out. When they try to move out, what happens is that there are certain receptors, stress receptors up here in the cervical region, my dear children, which becomes very much active. When these stress receptors become active, they are going to signal the the maternal pituitary gland to release a hormone that is known as oxytocin. What is this hormone? This is known as oxytocin. Okay, now what is happening? The baby is in over here. the baby tried to move out, then that's when it all happened. Apart from that, the placenta which is there, it is also going to release estrogen. And this estrogen, you know what it does? I told you we have endometrium, that is the glandular layer. We have myometrium, that is a muscular layer, right? On this muscular layer, the estrogen makes certain receptors active. What receptors, ma'am? Oxytocin receptors. Oxytocin receptors. Now the oxytocin which was released by the maternal pituitary will be coming and attaching over here, and when they come and attach over here, they will lead to what? Contraction and relaxation, contraction and relaxation, and that is how the baby will be finally moving out. Okay, so this is the whole process in there. Will you forget this? You should not be forgetting this. Okay, works. So fetal ejection reflex in humans triggers the release of oxytocin from the fetal pituitary. Is the baby's pituitary going to release oxytocin? No. Human chorionic gonadotropin from placenta. Human placental lactogen from placenta. Oxytocin from maternal pituitary. Yes. And what pituitary, ma'am? Posterior pituitary, not anterior. It is posterior pituitary. Always remember oxytocin and prolactin both by posterior pituitary, not by anterior one. Okay. Works. Anterior pituitary has FSH and LH and all these things being released by them. Cool. Do you have any doubts? With that, we complete human reproduction as well. But if you have any doubts, please ask to me, okay? If you're not asking, your doubts would not be solved as well. So make sure you are asking to me. I'll be really happy to help you out. Please go through the chapter once, pause the video, go through the chapter once and then come back to it. It will be really helpful for you to understand where I got the questions from. Yes? So, we'll take a break and we will come back soon. You go check out the video, not listen to me, just move the cursor out there. Yes? Okay champions, I hope you're back from the break and you studied everything out there. Let's get into the next chapter, reproductive health. So in context of amniocentesis, which of the following statement is incorrect? Okay, what do you mean by amniocentesis? First of all, you should be knowing about that. Suppose this is the baby, my dear children. The baby usually is inside a sac that is known as amniotic sac. Now inside this sac, there is a fluid which is known as amniotic fluid. Fine. So what happens is that on the baby's body, you know, it's made up of cells, right? And some cells, they shut down and they will be found in the amniotic fluid. Now the doctors will be taking out this fluid, yeah, and along with this fluid comes the cells out there. And with the help of the cells, you know, cells have nucleus will be having the genetic material, and by recognizing or basically analyzing that, we would be able to understand whether the child has any genetic disorders, because we are dealing with what? Genetic material, right? So we can test if the child would be having certain genetic disorders or not, or probably what is the sex of the child and all this information. So usually it was for finding out the genetic disorders, but now people started using it for what? For understanding the sex of the child, and if it was a female child, then they do female feticide, which is why it is completely banned in India now. So in context to amniocentesis, which of the following statement is incorrect? It can be used for detection of Down syndrome. Is Down syndrome a genetic disorder? Yes, my dear children, so I can find it. Now it can be used for detection of cleft palate. You might have seen people with cleft palate. Well, that is not a genetic disorder, my dear children. That is a structural abnormality. That is not a genetic disorder. So I'd say this is something that they can find out. But going ahead, it is usually done. This is something that is wrong. That is what I meant to say, yeah. And it is usually done when a woman is between 14 to 16 weeks pregnant. Is that correct? Yes, that would be safe zone for doing amniocentesis, correct. Now, it is used for prenatal sex determination. It is used, yes, it is used for prenatal sex determination, but it is legal. Is it legal in India? No, but elsewhere it is legal, okay. So I'll say this is also correct. So I'll say this one is the one that would be a wrong statement. Next, which of the following cannot be detected in a developing fetus by amniocentesis? Down syndrome, jaundice, Klinefelter syndrome, sex of the fetus. So you have to recognize first of all what are the genetic disorders, and basically if it deals with sex, all of it can be found out with the help of amniocentesis. So Down syndrome is genetic disorder, Klinefelter's is a genetic disorder. This obviously has to do something with the genes of the organism, right? But jaundice, it is not a genetic disorder, so which is why I say that I cannot find out whether the child has jaundice or not with the help of amniocentesis, because it is not a genetic disorder. Okay, so I can rule out B option. Going ahead to the next. Which of the following contraceptive methods involve a role of hormone? Okay, so we have many different types of contraceptive methods, right? What are those? Natural methods, yeah, where we are not injecting anything in our body. Basically, we're naturally preventing what the conception. For example, in a natural method, the first method that you have is coitus interruptus. Okay, you're interrupting between the sexual act, and before the man ejaculates, you're removing the penis from the female body. That is known as coitus interruptus or withdrawal method. Second thing, you have periodic abstinence. What you are doing in over here? Between a period, okay, so there might be some period, a fertile period where the woman is the most fertile, right? That is almost from 10 to 17 days of the whole menstrual cycle. During this period, the partners, they would not be having sex. This is known as periodic abstinence because it is the most fertile period. Again, are you using anything external source? No, which is why it is considered as a natural method. Now there is one more, lactational amenorrhea. What do you mean by that? Basically, a lactating mother would not be menstruating. Okay, because in her body the level of prolactin hormone is way too much, the one that helps in the formation of milk and things like that, okay, which is why it is way too much, and it inhibits the other hormones, that is gonadotropin releasing hormones like FSH and LH, which is why ovulation would not be happening. But this is not reliable methods. All these methods that I am talking about, they are not at all reliable because they are natural methods, right? The second comes barrier method. In that comes condom, walls, diaphragm and everything, where again you are not releasing any hormones and things like that. Now the third one that comes is what? IUDs, intrauterine devices. Again, you have three of them. Intrauterine devices, first of them is what? Is the non-medicated IUDs. That means if they are put inside our body, they are not going to release any medicinal stuff or things like that. They are just going to act foreign. They are just going to act foreign in our body. And when something foreign comes in our body, what will happen, my dear children? Our body would be reacting to it. Yes, so our immune system becomes active and they will react to it, but not in a way that we would be reacted as such, but just that particular area, which is why the sperms which would be entering, they will also be killed because your immune system is really active up there. Yeah. Now the second thing is copper releasing IUDs. What is happening over here? Basically, these IUDs, when they are inserted into the uterus, they are going to release some good amount of copper ions. And what are copper ions going to do? They are going to mess around the motility of the sperms. The sperms would not be able to move when there is copper ions. Yeah. And apart from that, they also do one more thing, that is fertilization capacity of sperm is greatly reduced because of copper T. Okay. Now, this is copper releasing IUDs. Third one is hormone releasing IUD. This is where you release hormones, right? So hormone releasing IUD as in progesterone, LNG20 and things like that, where they are doing one thing, that is in our body we have a level of progesterone and estrogen, right? Now they are giving synthetic hormones, and when we are giving synthetic hormones to our body, the level of normal hormones becomes disturbed, my dear children, which is why the normal function that we're doing, they would be doing the opposite of it. They would not allow ovulation to happen. They would, instead of making the endometrium thick, they will make it thin such that implantation would not be happening. And also the cervix which is there, they will thicken up the mucus up there so that this sperm would not be traveling. So this is hormone releasing IUD. There you need hormones, right? Now that was the third IUD thing. Okay, or what do you call it? It was the third method of contraception. Now talking about the fourth method is pills. In pills also you have hormones, same progesterone and estrogen combination, or simply them alone. Okay, you give them individually. Basically again they do the same function as I told you in hormone releasing IUD. They stop ovulation, or basically it does not allow implantation to happen, makes the cervical mucus thick and things like that. And apart from that, you have implants. Implants are put under your skin, my dear children. Again, they are also going to release what? Hormones. So these are the hormonal method. And the last one is surgical method, where you're going to cut the vas deferens, does not allow the transport of sperm, and you're going to cut the fallopian tube, does not allow the transport of ovum in there. Yeah, so over here, if we talk about everything, which are those things that releases hormones and prevents contraception? Pills, emergency contraceptives, barrier method. Barrier method is not going to release any hormones up there. Yeah, lactational amenorrhea, pills, emergency contraceptive. What do you think? Barrier method, lactational amenorrhea and pills. Barrier method is there, again ruling it out. Okay, CUT, pills and emergency. CUT releasing copper, not hormones. So I told you in lactational amenorrhea you have what? Prolactin hormone being released, which is why everything is being stuck. Pills, again hormones. Emergency contraceptives are also there, which are given, for example, someone is raped and things like that. So at that point of time, within 72 hours of coitus, you are giving this emergency contraceptive such that even if ovulation has happened, my dear children, even if fertilization happened, implantation does not take place. So actually it prevents ovulation also, but if it has happened, it cannot prevent it, right? So anyways, so all these things. Okay, so basically I have three things which has a role or involvement of what? Hormones in it. Now question number 4, which of the following approaches does not give the defined action of contraceptive? Hormonal contraceptive, vasectomy, barrier methods and intrauterine devices. Hormonal contraceptive, my dear children, what do? I just told you. What do? They prevent fertilization. That would be the best thing to explain. Okay, they prevent what? Fertilization. What about vasectomy? What about vasectomy? Vas difference you're cutting, my dear children? You're cutting the vas difference. So obviously, do you think transport would be happening? No. So we have to see. Prevents entry of sperms, prevents ovulation. Wait. I have to do some changes up here. Okay, let's start with this, because I think this would be very, very, very helpful. Okay, so over here, hormonal contraceptive goes with this only. Okay, barrier method goes with this only. This goes with this only. You are asked which one is the one that does not matches up. That is the question. I got messed up. Okay, so my dear children, vasectomy, it prevents spermatogenesis. This is completely wrong. Why do I say it's wrong? In vasectomy, what are you doing? You're going to cut the vas difference. Okay, you're going to cut the vas difference. So that means sperm is not able to transport. Sperm is not going. But what about the semen? Is it made or not? About the semen, it is made or not? So semen is somewhere over here, my dear. Semen is being produced by what? The seminal vesicle, the prostate gland, and the bulbourethral gland. So semen will be still produced. Okay, semen is still going to be produced. It's just that the sperm is not going to travel. Are the sperms going to be formed? Are the sperms going to be formed? Yes, spermatogenesis is going to happen, but just that the transport of sperm would not be happening. Okay, transport of sperm would not be happening. That is the only thing. Spermatogenesis will be happening, and also semen formation is also going to happen. Semen, by semen I mean seminal plasma formation. Semen actually includes both sperm and seminal plasma, but according to your NCERT, it says that semen is there but without sperm. Semen is there but without sperm. That is one very important question, which is why I spoke about both of them. Yeah. So in vasectomy, it does not prevent spermatogenesis. Spermatogenesis is there. It just prevents the transport of sperms. Cool. Now, which one of the following is the most widely accepted method of contraception in India at present? Cervical caps, tubectomy, diaphragms, or IUDs? It is IUDs. IUDs is one of the most widely accepted thing, and you know what? It is best for spacing children also. For example, a couple had a child and then after 5 years they want to have a child. So basically they can use IUDs. Okay, doctors would be inserting it and removing it according to the wish of the patients out there. So it will be IUDs. Next thing, given below 4 methods and their modes of action in achieving contraception. Select the correct matching from the 4 options that follow. Okay, the pill, condom, vasectomy, and copper T. The pill which is there, what does it do? The pill which is there, which is hormonal, my dear children, it is hormones basically, and I don't see any hormonal option over here, so I can directly say it prevents ovulation. So it's the hormones that prevents ovulation. Okay, so that's the only thing. So A goes with 3. A goes with 3. I have 2 options, so basically I can cut off the other 2 options. Okay, going ahead to the next thing. Condom. Condom, what does it do? It is a barrier method. It does not allow the sperm to move, right? So it prevents the sperm reaching the cervix. Again, that is correct. So B goes with 1. B goes with 1 over here. So I already got my answer, but let me see the other options. See, vasectomy. What happens in vasectomy? My dear children, I told you, semen is there, but it contains no sperms because sperms are not being transported. So C goes with 4. Okay, now what about D? Copper T, my dear children. Copper T would be preventing implantation. Okay, because it is not allowing a proper, say, copper T. Yeah, copper T. Actually, I cannot say copper T prevents implantation. It's a sub effect. Okay, copper T does not allow the sperm to move. Yeah, copper T does not allow the sperm to fertilize. So ovulation is not happening. So obviously implantation would also not be happening. So I won't say it is the correct method, but yeah, that would be comparatively correct. Okay. Going ahead, medical termination of pregnancy is considered safe up to how many weeks of pregnancy? So medical termination of pregnancy is almost safe up to 12 weeks of pregnancy. Okay, now I'll tell you one more thing. Medical termination of pregnancy, MTP, or induced abortion can be done up to 24 weeks as well. It can be done up to 24 weeks, but it is considered to be safe only up to 12 weeks. Got it? Yes. Now, select the option including all sexually transmitted diseases. Okay, only sexually transmitted diseases. Gonorrhea, syphilis, genital herpes. Gonorrhea, malaria, genital herpes. Malaria is caused by mosquito bite, not sexually transmitted disease. Okay. AIDS, malaria, again, malaria is there. AIDS is a sexually transmitted disease. What about filaria? Filariasis, my dear children. Filariasis is also transmitted through mosquito, Culis mosquito bite. Again, wrong. Cancer. Okay, cancer, AIDS and syphilis. Cancer is not a sexually transmitted disease, please understand that. So you have only one option, and that is this. That includes all sexually transmitted diseases. One is gonorrhea, one is syphilis, and the other one is genital herpes. Works? I hope it does. Next, match the following sexually transmitted diseases with their causative agent and select the correct option as well. So gonorrhea goes with what? Gonorrhea, syphilis, genital warts and AIDS. I'll tell you the easiest one, how you can find it. AIDS, so everyone know AIDS is caused by HIV. Call it HIV AIDS, right? So D goes with 1. D goes with 1. How many options do I have? 3 options. Let me cut down this option. Okay. Now talking about which is more easy for you. Genital warts, yeah, genital warts also comparatively common. Genital warts is caused by human papilloma virus. Okay, it is caused by human papilloma virus. Now, gonorrhea and syphilis is there. You have gonorrhea and syphilis. Gonorrhea is caused by Neisseria, and syphilis is caused by Treponema pallidum. Okay, so syphilis is Treponema, gonorrhea is Neisseria. Please remember a question that could be coming around any time. Okay, so at least these information is very much important for you. So I'll say A1 goes with 2, A1 goes with 2. I just have one option like that. This becomes my answer. Okay, now syphilis goes with 3. Agreed? Yes. C goes with 4. And D goes with 1. Next, in which of the following techniques the embryos are transferred to assist those females who cannot conceive? So over here it is strictly asked that basically embryos are being given. Okay, embryos are being transferred to patients who basically cannot conceive. So which are the techniques through which I can transfer the embryos, my dear children? You have ZIFT, zygote intrafallopian transfer, where what do you actually do is that you take the sperm and the egg, you fertilize in the laboratory condition, yeah, and now you have the zygote. The zygote obviously starts dividing, but if it's suppose up to 8 cell stage, okay, we call it an embryo actually. Now it is 8 cell stage, so basically once the zygote divides into 2, that means it's not a zygote anymore, it's an embryo. But here it has said ZIFT. Why? Because you're just taking cells which has 8 number of cells, not beyond that, okay, and you can put it in the fallopian tube. So this is the embryo only that is being transferred. Apart from that, suppose it is more than 8 cells. It is more than 8 cells, you directly put it inside the uterus rather than the fallopian tube, and that is known as intrauterine transfer. That is known as intrauterine transfer. Apart from that, anything that is given up over here, they are not the methods in which embryos are being transferred. So ZIFT and IUT is the only method. GIFT is gamete intrafallopian transfer. You are transferring the gamete. So for example, I am a woman, I am not able to produce enough amount of gametes. I am going to take it from someone else, and that gametes have been put up in my body. So that ovulation and everything, fertilization and everything can happen in my body, rest of the things, okay, production of it probably might not be happening in my body. So this is wrong. ICSI is intracytoplasmic sperm injection. Basically you have the zygote, you have the cell, egg cell, yeah, and you're inserting the zygote, sperm into that. This is known as intracytoplasmic. Inside the cytoplasm you are inserting the sperm, intracytoplasmic sperm injection. That is how you make the zygote up there. So that is also wrong. We are talking about embryo, right? And this option is also not there. So I'll say it's A. That is the only answer. Next, in case of a couple where male is having a very low sperm count, which technique will be suitable for fertilization? Gamete intracytoplasmic fallopian transfer. This is something that they made up. There is nothing like that. Listen children, over here it is mentioned that they have what? Very low sperm count. Okay, very low sperm count means probably I cannot rely on that person to do natural sex. Okay, so basically we can do one thing, that is artificial insemination. We collect enough number of sperms, yeah, and then we artificially inseminate. We artificially put it inside the body of the female, inside the reproductive tract of the female. That is artificial insemination. Now apart from that, if that is also not working, we can do one more thing. That is what? We take a sperm, okay, we take this egg from the women and we insert it there. That is known as intracytoplasmic sperm injection. So for children who are having confusion, that is artificial insemination, intracytoplasmic sperm injection, all make sense, ma'am. Low sperm count, both of them works. So we say both B and C are correct answers in this situation when it comes like that. But whatever you're sure of, go with that first. Okay, so I'll be very much sure of artificial insemination, my dear children. That could work, and also either one. Okay, either one. So both the answers are equally correct. Please remember that. Intrauterine transfer is not correct. Okay, intrauterine transfer, IUT, is for embryo transfer. It is not sperm. Okay, please remember that. So I'll say B and C are correct options. Now, question number 12. Which of the following statement is correct with reference to a test tube baby? Okay, so fertilization of the egg is effected outside the body. The fertilized egg, okay, fertilization of the egg is effected outside the body. Do you think so? Yes, my dear children. In the laboratory condition, the egg and the sperm is made to fuse. Now, the fertilized egg is then placed in the womb of the mother where the gestation is completed. Okay, this fertilized egg, which is basically talking about zygote forms the embryo, and this embryo is put down in the womb of the mother, that is the uterus of the mother, where the whole, cycle of formation of the baby would be taking place. Is that correct? Yes, that is also correct. Fertilization of the egg is effected in the female genital tract. Test tube baby, it cannot happen inside the female body, okay? It is then taken out and grown in the last test tube. Why will you do that? Who grows a baby in the test tube? Wrong. A prematurely born baby is reared in an incubator. No, my dear children. Fertilization of the egg and the growth of the embryo are effected in a large test tube. No. Test tube baby, because it is happening, the fertilization is happening outside the female body in the laboratory condition, which is why it is known as test tube baby, not that it happens in the test tube. Please understand that. Okay, test tube means laboratory condition, that is what it means. So fertilization happens in the laboratory condition, and again it is put down in the female body. Only in the female body you get all the important nourishment for the baby to grow. Okay. Yes. Question number 13. Consider the statements given below regarding contraception and answer as directed thereafter. Medical termination of pregnancy during first trimester is generally safe. Is it correct? Correct. Generally, chances of conception are nil until mother breastfeeds the infant up to 2 years. You're talking about lactational amenorrhea, but I already told you, actually it's working up to 6 months. Up to 6 months, it is comparatively working, but that is also not very reliable. It is not at all reliable method of contraception. Please understand that. Over here, 2 years have been written. That is totally wrong. Now, intrauterine devices like copper T are effective contraceptives? Yes, it is, for sure. Contraception pills may be taken up to 1 week after coitus to prevent conception. Wrong. It can be taken up to 72 hours. Up to 72 hours, my dear children. Not above that. Okay? There is no need of that. Fine? So that is also wrong. So I will say 1 and 3 are correct. Which of them are correct? 1 and 3 are correct. Okay? Yes, that's all. So these were the questions of reproductive health, my dear children. Again, as I always tell you, if you have any more questions, okay, you saw somewhere you are not able to solve it by yourself, please put it down in the comment section. I am here to help you out with that. And if you liked the video, do not forget to like, okay, and also share with your friends. Also subscribe the channel, tell them to subscribe. We are planning big long-term things for you guys, okay, for the next year. So make sure you stay tuned with this particular channel. It will definitely help you out really well. Yes, so that's all. Bye bye, take care.